HIV is human immunodeficiency virus. HIV is classified as a retrovirus. Structurally, HIV takes a rough spherical shape. The retrovirus contains two positive single-stranded RNA housing the virus's genome, which is contained in the cone-shaped protein capsid. The function of this capsid is to protect the RNA from being digested by its own nucleases. This capsid also encloses other important enzymes and proteins such as reverse transcriptase. The phospholipid membrane and the viral protein matrix form the outer shell of the HIV virus. This shell encases the glycoproteins which are responsible for the attachment and transmembrane transportation of the virus. Unlike other viruses, a retrovirus uses the enzyme reverse transcriptase to produce DNA from the virus's own RNA, and then replicates by incorporating its integrated DNA into the host cell's genome to be replicated. Reverse transcription of HIV is highly unstable due to the lack of the DNA polymerase, which corrects errors made in replication, and this can create various mutations of the HIV strand. Some defective strands that cannot replicate, while some can become very resistant to antiviral and antibiotic medication. Antiviral medication works by limiting the virus's growth and development. One antiviral drug used to manage HIV is a protease inhibitor, which inhibits the cutting and assembly of the viral DNA. But as stated earlier, the unstable replication of HIV leads to many strands of HIV becoming resistant to these antiviral drugs. Due to HIV being a viral disease, antibiotics cannot treat it directly. The significance of the antibiotic is that it is necessary for HIV-positive people to take them because the immune system becomes compromised and therefore ineffective in fending off everyday infections such as salmonella. HIV limits the lymphocyte recognition, depressing the body's effectiveness in fighting and memorizing the invading pathogen, which can lead to multiple attacks by the same infection or disease. There are two types of HIV, HIV-1 and HIV-2. HIV-1 is more common, comprising of 90% of the known cases. HIV-1 is comprised of 4 subtypes, as depicted in this diagram. HIV-1 is a relative of viruses found in chimpanzees and gorillas in West Africa. The 4 subtypes include the following groups: Group M, Group N, Group O, and Group P. Group O is restricted to West Africa, and Group N is a strain that was discovered in Cameroon in 1998. HIV-1 Group P is closely related to gorilla simian immunodeficiency virus. More than 90% of HIV infections are HIV Group M infections within Group M. There are 9 genetically distinct subtypes. These 9 subtypes are Groups A, B, C, D, F, G, H, J, K, and CRFs. These various subtypes are distinguished by the region where they can be located. The subcategory CRF is significant because it indicates resistance to HIV. Group M is further divided based on the genetic sequence data. Circulating recombinant forms are derived from recombination between viruses of different subtypes which are given a number. HIV-2 viruses are less virulent than HIV-1, but it is still known for causing AIDS. Triple cocktail therapy is a combination of 3 antiretroviral drugs that are used to slow the progression of HIV. These medications are 2 nucleoside analog drugs such as AZT and 3TC, and a protease inhibitor such as Crixivan. The drugs drastically reduce the concentration of viri in the bloodstream to undetectable levels by affecting enzymes in the virus itself. Drugs do not completely eliminate every virus in the body, and probably never will. AZT, which is short for zidovudine cocktail, is a drug used to fight HIV and AIDS since the 1980s, and is a reverse transcriptase inhibitor. 3TC, which is short for lamivudine, is a nucleoside analog reverse transcriptase inhibitor. AZT and 3TC combined form Combivir, the brand name pharmaceutical treatment for HIV infection. In the 1990s, protease inhibitors, which target viral assembly by inhibiting the activity of transcription, were combined with reverse transcriptase inhibitors to form a cocktail which reduced highly active antiretroviral therapy, HART, to undetectable levels. HART are the aggressive treatments used to suppress HIV replication and progression. The original HIV infection attaches to the host and then replicates. Due to the unstable replication of reverse transcriptase, a drug-resistant, mutated strain of HIV survives the antiviral medication. That now resistant strain infects another host and then replicates. A new drug can be synthesized to kill the previously resistant strain of HIV, but another mutation can occur in the replication that is resistant to the newly synthesized drug. This process is why the triple cocktail therapy can drastically reduce the number of HIV molecules in a patient's body, but cannot completely treat the patient due to the constant replication and mutation of drug-resistant strains. Due to the rapid mutations of HIV, the mutated strands are more resistant to the antibiotics than the previous generations were. Therefore, the administration of triple cocktail therapy needs to be constantly revised to maintain its effectiveness.